While explaining the overview of carbohydrate metabolism in my previous lecture, I pointed out the how the glucose uptake is taken into the cell, and then the way how that is further processed and breakdown taking place, particularly talking of the aerobic glycolysis, in which pyruvate is the end product. Pyruvate is the end product. Here I'm pointing out that is the end product, occurs in cell with mitochondria and an adequate supply of oxygen. There is another type, that is the anaerobic glycolysis, in which the lactic acid is produced, and it occurs in cell that lacks mitochondria. Lactic acid is produced in those cells where there is no mitochondria, or in cells deprived of sufficient oxygen. So it's very clearly now talking on this issue that aerobic glycolysis where oxygen is available, and the anaerobic glycolysis whereas there is no oxygen available, and or insufficient oxygen is present. Then I also mentioned that glucose is transformed across the membrane by 14 glucose transporter isomers, which are known as GLUT1 and up to GLUT40. So I also told you that they are specific in their action. The GLUT1 is abundant in erythrocytes and brain, whereas GLUT, which is insulin dependent, is found in muscle and adipose tissue, and GLUT is found in liver and the beta cells of the pancreas. Now, if we proceed further, this is the layout in the map, as I told you, that in the case of the aerobic glycolysis, there is the pyruvate is the end product, and I told you in my previous lecture that pyruvate, when formed, it is now staged. It is the stage set for further breakdown, and pyruvate itself has got the it can be converted into four different ways. It will be converted. On one hand, pyruvate, as I told you, will be going into lactate formation in the absence of the oxygen. When pyruvate second decarboxylated, and after decarboxylation, it will be producing the acetyl-CoA. Here it is the acetyl-CoA. Now here is the carbon dioxide leaving. It is decarboxylation of the pyruvate and then producing acetyl-CoA. Pyruvate can also, when carboxylated, it will be producing oxaloacetate. And oxaloacetate is one of the component of the Tca cycle. And fourth, pyruvate can, when it will be decarboxylated in the presence of thiamine pyrophosphate, it will be converted into ethanol. So there is sometimes university question that what is the fate of pyruvate formed in glycolysis? So when asked like that, you have to answer that pyruvate got four chances to be converted in different forms. One, lactate formation when the oxygen is deficient or less oxygen is available, or when decarboxylated, it will be producing acetyl-CoA, and similarly, when carboxylated, it will be producing oxaloacetate and also can produce the ethanol. This is the fate of the pyruvic acid when formed. After giving you this background what I already discussed with you, now I would like to proceed further, and that is the Tca cycle, tricarboxylic acid cycle. It is here, it is Tca cycle. The book has mentioned it in very bold way that pyruvate that is converted into Tca cycle, and then the cycle reactions going on. Before going to these reactions and Tca cycle discussion, I would like to give you that what is the significance of the Tca cycle. What is the importance? What is the biomedical importance of Tca cycle? So it is also known as the Krebs cycle. When we talk about the importance of this cycle, it is the final pathway where the oxidative metabolism of carbohydrate, amino acids, and fatty acid converge. Now, I'm speaking the word converge. So, it is very clearly mentioning here that all the metabolisms, they are converging at this place, being converted to then carbon dioxide, and then also going for complete oxidation, and finally producing ATP. So very important it is the biomedical importance or the importance of Tca cycle, that all the metabolic, that is all the metabolites, that is carbohydrate, amino acids, and fatty acids, they converge at this point, and ultimately the purpose is the complete breakdown and at the end production of ATP. The cycle occurs totally, the second point I'm also telling, the cycle occurs totally in the mitochondria, and therefore is in close proximity to the reactions of the electron transport. Already we have discussed when talking of the electron transport chain that it is very clear, near to that, and this is how it helps produce the ATPs. The Tca cycle, the other point, is an aerobic pathway. It is an aerobic pathway because oxygen is required as the final electron acceptor. Reactions of different reactions, they are taking place in this Tca cycle, in this pretext. The citric acid cycle also supplies intermediates for a number of important synthetic reactions. If you look now all this cycle, it provides different intermediates for the other metabolic reactions. On one hand, there is the converging of all the carbohydrate, amino acids, and protein, and it also provides the intermediates. This is very important to understand. For example, the cycle functions the formation of glucose from the carbon of some amino acids. I told you in my previous lecture that when the case of the starvation, long starvation, because if in the small starvation, first the glycogen come in action and glucose is provided from that. There is breakdown of the glycogen and glucose is being provided, and but there is long starvation, then amino acids, they come into action and they provide glucose, and this happens through this cycle. So this cycle is important to provide glucose in the case of the deficiency of the glucose or in case of the starvation. Similarly, it provides building blocks for the synthesis of some amino acids and E. So don't take it as just a cycle, closed cycle, but it is an traffic circle. It is, you can call it the traffic circle. Now, very interesting that pyruvate going to the acetyl CoA, the pyruvate, this has been bold in this diagram. This is given on page number 109 of your textbook of Lippincott. You can open your book and you can also see there that the pyruvate is converted into the acetyl-CoA. Now this conversion is very important to understand. It is not simple as you are finding in the diagram or in the chart. It is not like that. So once the pyruvate is formed, it must be transported into mitochondria before it can enter the Tca cycle. So I repeat this, that once the pyruvate in the case of the glycolysis is formed, it must be transported into mitochondria before it can enter the Tca cycle. And this is accomplished by a specific pyruvate transporter that helps the pyruvate across the inner mitochondrial membrane. This is how it is transported. Now, the transport of the pyruvate into acetyl-CoA, there is involvement of the multiple enzyme complex. It is not one enzyme, there are multiple enzyme complex. And we call this complex, no, I repeat, we call this complex as pyruvate dehydrogenase complex, which is a multi-enzyme complex. It is not the part of Tca cycle properly, but is a major source of acetyl-CoA. So what we need is the acetyl-CoA formation before proceeding to Tca cycle, my topic under discussion today. And this conversion of pyruvate to acetyl-CoA is under the influence of the pyruvate dehydrogenase complex. Why we call it complex? It is because now this is the pyruvate dehydrogenase complex. Now you see this pyruvate dehydrogenase complex is aggregate of three enzymes. I call it complex because there are a number of enzymes involved here. And each enzyme has got its importance. The deficiency will be creating certain disorders. For example, the deficiency of E1, which is mentioned here, will be creating, I'll let you know later on what disorders there will be taking place there. Now, here it is only to understand is that this pyruvate dehydrogenase complex is aggregate of three enzymes. What are those three enzymes? Number one, pyruvate dehydrogenase, or you can call it as E1. Number two, dihydrolipoyl transacetylase, or E2. And third one is dihydrolipoyl dehydrogenase, or E3. So what the author has categorized it, that is E1, E2 or E3. But you must remember what is E1, that is the pyruvate dehydrogenase. E2, what is that, is dihydrolipoyl transacetylase, and third one is dihydrolipoyl dehydrogenase. These are three the enzymes there, but in addition to the complex also contain two tightly bonded regulatory enzymes. I'm giving stress here that there are two regulatory, the word itself is very meaningful, regulatory enzymes. They help regulate the reactions. And what are those enzymes? And they are the pyruvate dehydrogenase kinase. Very important. When we're going into detail, you will see what role the pyruvate dehydrogenase kinase is playing and what's happened when the deficiency of this enzyme is there. It is a regulatory enzyme. And second one is pyruvate dehydrogenase phosphate. Now, this is the total number of the uh, enzymes which are present in the dehydrogenase, pyruvate dehydrogenase complex. Sometimes there is university question, mention the enzymes which are present in this PDH, pyruvate dehydrogenase complex. So you have to answer that by one, how I have uh, mentioned here in this diagram. In addition to these enzymes, there are certain coenzyme also. You see, we are calling it a complex. What does it mean complex? There are many number of things there, so that is why it's known as complex. There are coenzymes also. Early on the first slide, I showed you the number of enzymes. There are three: E1, E2, E3, and then two regulatory enzymes. But there are also present coenzyme. And PDH coenzyme, PDH complex coenzymes, that act as a carrier of oxidant for the intermediates of the reactions. For example, now you can understand it. The E1, with what I explained in the first slide, that requires thiamine pyrophosphate TPP. If that is deficient, then E1 will not be working and there will be certain disorders. Similarly, E2 requires lipoic acid and CoA. E3 requires FAD and NAD. So these coenzymes are very important to facilitate the functioning of the main enzyme which I have mentioned in my slide before this. I talking the deficiency of thiamine. So you can say that deficiency of thiamine or niacin can cause serious central nervous system problem. Now I'm just quoting here one example to you. You can well understand that the deficiency of one thiamine or niacin can cause a serious central nervous system problem because now this is because brain cells are unable to produce sufficient ATP via Tca cycle. Now it will make you very clear now here that how these coenzymes are important. Deficiency of one that can lead to these serious central nervous system problems. Now, this complex is very important from examination point of view and to understand the further proceeding of the Tca cycle. Coming back, then again, I'm talking of the conversion of pyruvate to acetyl-CoA, and there I was talking to you the involvement of a number of enzymes because to convert the pyruvate to acetyl-CoA is under the influence of number of enzymes and these are not part of Tca cycle but they are there, and in that pretext I will give you another example, that is as I told you, the pyruvate dehydrogenase. In my previous chart I showed you, and pyruvate dehydrogenase deficiency, what will happen? This is another example I'm putting. And that pyruvate dehydrogenase is E1, also known commonly, of the PDH complex. It is not very common, but because of although it is rare and is not most common, biochemical cause of the congenital lactic acidosis. Now, this enzyme deficiency results in an ability, I stress the word, this enzyme deficiency, that is pyruvate dehydrogenase enzyme deficiency, results in an inability to convert pyruvate to lactate. Here. So if there is problem in E1, that is pyruvate dehydrogenase deficiency is there, E1 deficiency is there, it will be not properly converting the pyruvate to acetyl CoA, and there is chances of this then the lactic acid formation is there. This enzyme deficiency results in an ability to convert, I'm repeating it, to the acetyl CoA, causing, no important thing I'm talking, causing pyruvate to be shunted to lactic acid. Clear? So if there is deficiency of E1, there is that conversion of that will be not taking place, and lactic acid formation will go on forming there. This is a disorder then. And this will then cause particular problems, the lactic acid production for the brain, which relies on the Tca cycle for most of it. Now I will go to the E2. First I was talking of E1, what I talked to in the complex. Now I will give one very clear-cut example of E2, and that is, in that pretext, in that reference, I will call it the arsenic poisoning. It is important to know arsenic poisoning question in the university paper is also many times. So, or when discussing the glycolysis, there you have already gone through that what happens when arsenate is there, arsenic poisoning, what happens that though the process of glycolysis continue, but the ATP production is restricted. So that is, in brief, I'm giving you, that is the how arsenic poisoning is taking place. Now, arsenic can interfere with glycolysis. Now I'm talking of this glycolysis where the poisoning is taking place, chances of taking place at the glyceraldehyde 3-phosphate step. And what will happen? Decreasing ATP production. As I told you, there will be decreasing of the ATP production. Arsenic poisoning is, however, due primarily to inhibition of enzyme that requires lipoic acid, the coenzyme I mentioned before this, that in that my diagram to you, that lipoic acid deficiency. I repeat it, arsenic poisoning is primarily due to inhibition of enzyme that requires lipoic acid as a coenzyme, including E2 of the PDH complex. So this is another example of what I was talking to you. As a result, the pyruvate and consequently lactate that accumulates, like pyruvate dehydrogenase deficiency, this particularly affects the brain, causing neurological disturbances and death. So this indicating the importance of these coenzymes, and this is what I'm talking of the arsenic. I would like to Now, tell you a few very important things. In your book, at the end of the chapter, very some nicely prescribed summary of the chapter is given there, and then there is a chart, there is a diagrammatic presentation of the whole process of the carbohydrate metabolism breakdown and in glycolysis. Here it's mentioned in detail. If you just go on moving it, you will be finding what we have studied. What you have to do is now with that, and every time when you finish a chapter in the book, at the end of the chapter, the summary of that chapter is given. Why I'm telling you this thing, that you must go through this summary, because most of the MCQs which are in the university questions, they are picked up from these summaries. So it is not only for this chapter, I'm saying for every chapter, for every thing. When you go for biochemistry, you must study, you must go through the summary given at the end of the chapter, and then the chart presentation, because it is also clearly indicating and telling you the whole process, and it will be giving a complete picture in your mind that how the process is going on, in particularly in the case of this glycolysis and Tca cycle. I'm available for your question anytime.